My work constituted of the discovery of transposable genes and my theory of genetic transposition.
I remember. It was something like jumping genes.
Yes, that was what my work and studies until 1930 amounted to. Instead of jumping genes, I termed it genetic crossover. In 1931, I discovered linkage groups in Drosophila fruit flies. Much of my work was concentrated on maize, and I explored its chromosomal, morphological, and evolutionary characteristics. The main focus was on knobs analysis, or the swelling of chromosomal material in certain positions. I also coined the terms of dissociator and activator, which are two new genetic loci. They're the gene controllers that dissociate or break chromosome and only work together. Hence, due to the transposition or change in position of the gene, it was what I defined as a controlling element. Sadly, in 1950, I was acutely aware that my work was at such a different level from the common wisdom of the time that I dared not publish an account of my research. It was such a completely new concept with such extensive implication that suggested an altered concept of gene mutation. I didn't want to make any statements until the evidence was conclusive enough to make me confident of the validity of the concepts. I needed to determine how this controlling element applied to the genetic systems of other species and how they regulated the genes. It wasn't yet understood how complex organisms underwent cell differentiation with the same set of genes.
Although, in 1981 you published The Chromosomal Constitution of Races of Maize. This became a national landmark that significantly contributed to evolutionary botany, ethnobotany, and paleobotany.
Yes, I did, with Blumenschein and Cato.
As I've repeated before, my influence was that I first sequenced the human genome. Not only that, I used my specially developed technology of express sequence tags, ESTs, to help decode different gene sequences, including the one responsible for colon cancer.
My significant contributions mainly revolve around the improvements I made using X-ray diffraction and crystallography. I photographed clearly the DNA structure using X-ray crystallography in my famous photograph 51 of the wet and humid form of the. As well, I refined the X-ray machines for better exposure. In my 35 years of life, I published nearly 50 scientific papers, 5 of them on my coal and charcoal natures and efficient usage before I was 26, and 17 on structural virology of tobacco mosaic viruses during the 5 years that I worked on that research. However, I am simply content with creating the method of analyzing complex, organized matter using X-ray diffraction and crystallography instead of simply crystallized solid images of specimen.
J. This is why you are considered the Dark Lady of genetics, though you didn't receive much recognition, partly due to me and Crick. However, John Desmond Burnell, one of the UK's most renowned and controversial scientists and X-ray crystallography, praised that you were distinguished by extreme clarity and perfection in everything you undertook. Your photographs were among the most beautiful X-ray photographs of any substance ever taken. Their excellence was the fruit of extreme care in preparation and mounting of the specimens, as well as in the taking of the photographs.
Is that so? How kind of him.
The work I did was when Crick and I created the historic model of DNA, with its double helix on the outside made of a phosphate and sugar group backbone, the latter like rungs of the base pairs inside, and the anti-parallelism of the two strands. On my own, I theorized about virus structure, and studied X-ray diffraction in RNA as well as its role in protein synthesis. When I became the director and president of the Cold Spring Harbor Laboratory in New York, I mainly assisted in the studying of tumor virology, the molecular basis of cancer and oncogenes or cancer genes.
On the same topic, can both of you tell me more about the great DNA race?
I wouldn't quite term it a race.
What started it mainly was, in 1950, the gene was considered the smallest unit of genetic information, and it was unknown what it looked like structurally, chemically, and how it was copied.
My approach was to gather data from my X-ray diffraction, study the images, and then build a model with the conclusions I drew.
Our plan was to make a model, and then narrow down the different possibilities. We decided to use our colleagues' experimental results, our own intellects and resources, and persistence. In the end, we took the ideas of Alexander Todd, Erwin Chargaff, Maurice Wilkins, and you, Rosalind Franklin, as well as Linus Pauling to make our model. It all fell into place when Jerry Donahue, a physical chemist, pointed out the incorrect ring configuration of the rings in the TNG bases. From there, I made some cardboard cutouts on February 28, 1953, had a eureka moment, after some rearranging, and then we were done. We published a one-page paper with the understated title of A Structure for Deoxyribose Nucleic Acid in Nature April 25, 1953, with a schematic drawing of the double helix by Crick's wife, Odile, and simply tossed a coin to decide the order of authors. I'm sorry to say, Rosalind, we completely disregarded you, and held patronizing attitudes towards you. I believe that reflected on the contemporary nature conventions of gender and science during that era.
That's fine. I readily accepted the model and still published my work. In the end, we became friends and I even spent the last period of remission in Crick's home. I enjoyed my work and I don't regret anything.
All right, I'll accept that. Next is, did Wilkins really backstab you, Rosalind?
I don't believe he did. If anything, I think his frustration that he couldn't develop his idea on how NA was shaped, and his model failed, might have led to his demise and his incorrect choices.
At the time, we thought Wilkins had your consent to show the work to us, the MRC report from Max, as well.
What's past is past. It can't be helped. What's more, if Randall hadn't omitted so much when he invited me to join the project, it would have caused less misunderstandings.
It might have also been your attitude, you know. Wilkins was quite reticent while you're quite intimidating.
It's simply a manner of how I was bred and raised. One didn't complain. One dealt with it.
That is so fascinating. Last question, Venter. Since I hear you're such an egoist, you'll enjoy this question. How did you map out the complete human genome?
Lad, it was a very long and gruesome process. Before I started my formal education, I did a tour of duty as a Navy corpsman in Vietnam from 1967 to 1968. See, this is what I looked like. Then when I got back, I began my education. By the time I was done, I had developed a new technique to identify unknown genes called express sequence tags, or ESTs for short. These are used to help decode the gene sequences, so they can be organized and mapped much more easily. I heard you tried to patent gene fragments later on with your technique. That seems quite controversial. I truly began to sequence genomes in the early 1990s. In 1995, I determined the genomic sequence of Haemophilus influenzae, which is the bacterium that causes meningitis. Then I moved on to various other sequences, like mice and rats. Finally, I joined in the Human Genome Project with my colleagues, and you completed the sequencing in 2000, right? I remember the article on February 16, 2001, in the Science. You also appeared on the cover of Time magazine on July 3, 2000. That issue, what was it called again? Yes, Cracking the Code. Actually, no, that was simply the rough draft. It was actually fully completed in 2003. Then in 2007, we announced the first successful mapping of the complete 6 billion letter genome of an individual human. Ahem, yours truly.
Father, this man is such an egoist.
I concur with you, Mendel.
Scuffs, hey now, I'm not an egoist, I'm an egomaniac. You see here, I wouldn't have mapped the human genome if I was lacking in confidence. Not only did I map out the human genome, I founded multiple companies and organizations to help with genomics research, including Celera Genomics, the J. Craig Research Institute, Synthetic Genomics Inc., and so on. My goals with these companies are to commercialize genomic-driven solutions and address global needs, as well as continue to develop innovative genomic tools. Last but not least, in 2010, I worked on the project for synthetic DNA, where we produced the first man-made bacterial cell, Mycoplasma mycoides V0 sign 1.0 JCV, and it was named after me. We combined it with a synthetically designed genome, where it created proteins according to the synthetically designed genome.
always so boastful.
That is true.
Continues mumbling. And then I wrote more than 250 research.
That was really satisfying. I don't have any more questions, but maybe you do. I hope I can answer them, though.
Child, I have a simple question that I believe all of us are wondering.
Well, basically, I'm at a psychic's abode, Madame Kismet. She summoned you here to this metaphysical realm so that I can have a chat with you guys. She can even summon spirits of people who aren't dead, which is why you two, Watson and Venter, are here. It's astral projection.
Outrageous. What the heck is this astral projection? I'm a young man. I may be old, but not that old. What are you up to? This is not science.
I do not understand, however. I am quite through with this meeting. Can we call it adjourned?
I quite agree. All this associating is too much. It's too crowded and noisy.
Explain. Astral projections, rubbish. It's just hocus pocus.
She did say not to anger the spirits, so I better stop now. Thank you, spirits. Alright then. So I simply close my eyes, count down from 3. 3, 2, 1.
Welcome back. Did you enjoy your excursion into the astral realms?
Whoa! I'm back! That experience was beyond... I'm speechless. Thank you so much. It was truly enjoyable, and totally worth the 5 years of being a slave. Hopefully I can experience it again someday.
We'll see about that.